Today I will be presenting a slideshow based on the work I have done over the summer through NASA's N3 internship program in collaboration with Dr. Priya Dasarma. Over the summer, I researched the purple Earth hypothesis in order to find a way to explain this hypothesis to the general public. In order to present information in a more accessible way, I chose to research the purple Earth hypothesis and use my new understanding of the topic to present the information in a more visually enticing way with less complicated wording to help the information be more accessible, easy to understand, and interesting for the general public. This is because the public typically sees scientific articles as too wordy or confusing to read and actually gain the information from. Over the summer, I hypothesized that potentially using visuals and less complicated wording would help make that information more accessible and understandable for the public so that the information on new scientific discoveries can be shared with everyone. I will now be showing you the presentation of information that I had created over the summer to spread this information in an easier and more accessible way. Current research hypothesizes that early planet Earth was visibly purple. But why? The purple Earth hypothesis is a space-centered biological hypothesis first proposed by molecular biologist Priya Dasarma in 2007. PEH states that before there was oxygen on Earth, a special microscopic organism existed. This microscopic organism absorbed green light from the sun and reflected purple light, thus giving it a purple color. These organisms are able to survive without oxygen. Thus, before there was oxygen on Earth, large amounts of these organisms may have made the Earth's surface appear purple. Those microscopic organisms are Haloarchaea, or more specifically, Halobacterium species NRC1. A haloarchaeon is a type of ancient, microscopic, single-celled organism that thrives in extreme biomes, specifically extreme salt biomes. NRC1 can even do so without oxygen. NRC1 is considered the model microbe for Haloarchaea, due to it being extremely simple to culture and being one of the first organisms to have its entire genetic sequence mapped out. But how could this make early Earth purple? The answer lies within its cell membrane, specifically a special part of its cell membrane called the purple membrane, where light from the sun is turned into usable energy in the simplest way currently known. The purple membrane is a specialized region of the membrane that contains a 2D lattice of a protein called bacteriorhodopsin. This acts as a light-dependent proton pump that spans from the inside of the membrane to the outside. This pump is used to create and maintain a concentration gradient, where one side of the membrane has a different amount of molecules, in this case protons, than the other. The pump for the concentration gradient is powered by green light and pumps the protons outside of the cell membrane so that there are more outside the membrane than inside. Protons will move towards the inside to even out this gradient. However, their only way of getting inside is through a protein called ATP synthase, which uses the protons going through it to create energy in the form of ATP. BR uses the purple pigment retinol to collect green light and use that light to pump ions outside of the membrane. Then ATP synthase spins as the protons diffuse back into the membrane through it and attaches a phosphate ion to an ADP molecule to turn it into an ATP molecule. This ATP can be used for energy throughout the cell. Due to the simplicity of the process the purple membrane uses, part of the PEH hypothesizes that the purple membrane predates photosynthesis as we know it now. In fact, it's also hypothesized that photosynthesis emerged to counter the purple membrane, as the pigment used for photosynthesis absorbs light on either side of where retinol absorbs light on the spectrum. The process that the purple membrane is part of uses a pigment that absorbs the most abundant color of light on the spectrum, green, NRC1 uses retinol, which absorbs green light and reflects pink and purples, causing the pink and purple color they have, while most plants now use chlorophyll, which absorbs light on either side of the spectrum from where retinol does, making them green and yellow. This is how scientists got the conclusion that photosynthesis likely evolved after NRC1 as a competitive measure, since NRC1 can float to the surface to absorb the most light. Why is any of this important? Well, here are a few applications. In terms of biotechnology, the key protein in the purple membrane can be used in artificial retinas. The gas vesicles that NRC1 uses to float can be used in vaccine development, and some types of haloarchaea produce what is considered an alternative to plastics, and many more applications. In terms of biosignatures, NRC1's use of the purple membrane to turn solar energy into chemical energy is an incredibly simple method of energy transduction that suggests it could evolutionarily be one of the first, as it is the simplest method known at this time. In terms of life in space, prior to the knowledge of haloarchaea, the search for life on other planets was confined to planets with blue or green color, as it was thought to indicate chlorophyll. However, with the knowledge of retinol as another pigment that primary producers may use, the search could be expanded to include planets of red and purple colors as well. In summary, not only did this project teach me about a hypothesis I had never heard of before, but it also helped me grow my understanding of how to read and interpret scientific articles and how doing so is not common practice for the general public. To most people, scientific articles are like trying to read Shakespeare. Hopefully, my hypothesis of presenting information in other ways with visuals, more simple words, and less text at once can lead to the public having significantly more understanding and knowledge on new discoveries, as well as allow the information to be more accessible to others in different aspects of the scientific community, allowing more future collaborative labs, hypotheses, theories, and discoveries to be made. Works that I have cited throughout my presentation. I would like to thank Dr. Priya Desarma for helping me through this project as my mentor, as NASA's N3 internship program for giving me the opportunity to have this presentation and research what I have over the summer. Additionally, I would like to thank everyone attending the presentation for listening to my talk. Are there any questions?